Okay, so the lecture for the week is on chapter 6, which is the phylum Porifera, the sponges. So we're moving from our discussion of the creation of diversity and the study of diversity, how to recreate the evolutionary history of groups, how to group them, how to name these different species, what a species is. That's our sort of module 1. Module 2 begins with looking at animals phyla by phyla. Um, and the first phyla is phylum Porifera. All right, this is just a example of a species of sponge described in 2019 in the journal Zootaxa. Bring this up just, we've been talking about how to name a new species, describe species, and just want to reiterate the fact that this is happening all the time. New species are being defined. Also get you guys comfortable with looking at citations of journal articles. So here are the authors: Tabachnik, Fromont, Ehrlich, Menshaniana, right? Published 2019 in the journal Zootaxa. Hexanellida from the Perth Canyon with descriptions of 5 new species. So this is in the phylum Porifera, class, order, family. The genus is Calyptotoreti and Falcorae. This is what it looks like. This comes right from the species description. This is the type specimen. It shows the spicules, and this is one of the characteristics that is good at describing Calyptoreta falcorae. So I think this was named after a guy that owns a marina and was really generous to these scientists. So phylum Porifera, some famous members of this phylum. Okay, so we always start with this sort of broad overall view of the phylum. What we find here, the sponges, is a very ancient group, Precambrian, so more than 600 million years ago is when sponges came into being. So this really was first animals. Okay, despite them being around for a very long time, there's only a little more than 5,000 species that have been described. Where do you find them? In the ocean, but there are actually a few species that are found in freshwater, including here in Georgia. And as to their ecology, they are sessile or non-moving as adults, and their larvae are what we call planktonic. And plankton just doesn't mean it's not a specific taxonomic group. It's not a family or order or anything. This is a description of it's really a description of their niche. Planktonic means that you float on the ocean currents. Okay, so adults stationary, plankton floating around, dispersing from the adult, settling down someplace. One of the most remarkable thing about this phylum is the fact that they do not have any true tissues. Of course, they have cells. Everything that's living has cells. Cells are specialized into a few different cell types, but those cell types don't really aggregate into what we could consider to be tissue. So these are very simple organisms. Some species of sponge have these symbiotic algae. These are usually protists that are photosynthetic. They live in the tissue of they live in the cells of the sponge, and they get a place to live, and the sponge gets some extra sugar that is produced by the algae. How do they make their living? Basically, most of their energy comes by filter feeding. Okay, so they filter out a massive amount of water every day to get the dead stuff, detritus, and then also microorganisms from the water. And we'll talk about how they accomplish this with their really simple design. And here's some pictures, lots of diversity in terms of form. The color usually comes from the algae that lives in the sponge. Porifera, think of pores. See all these little holes and openings in them. That's kind of what defines a sponge's body, is those pores. In terms of their morphology, three main types of cells. Those of you guys who've been to lab, these will sound familiar. The pinacocytes. So cyte means cell. So we have the pinacocytes, which are basically the covering on the outside of the sponge. Choanocytes do most everything, right? They are They have a flagella or a whip-like tail, and we see that they are responsible for generating the current. They're also responsible for filtering the food out of the water, and they even produce gametes, right? So these gametes are the sperm and the egg. They are a sexually reproducing organism. We'll look at these. Choanoflagellates are another group of organisms. They're not animals. They are what we call protists. And so we see this as being a potential origin of sponges, was a group of choanoflagellates living together could have formed what becomes the first sponge. And then the third type of cell, these things called archaeocytes, or sometimes called amoebocytes. And they, like an amoeba, move around in the body of the sponge. They distribute nutrients, waste. They actually make the spicules or the skeleton, and they can be involved with asexual reproduction. All these little things called buds or gemmules. So here we have pinacocytes on the covering. Here's the nucleus. Show you it is a cell type. Here are the choanocytes with their little flagella, and then here are the archaeo or amoebocytes. Mesohyl. This is basically a collagen type matrix that is secreted by the animal, and that is when you see a sponge at an art store or something, that's really what you're looking at, is this mesohyl. Spicules are embedded in the mesohyl, and they're there for both support and protection. So here's some of those choanoflagellates. Okay, we have their sessile attached to the substrate. They're single-celled. They have a flagella, and if you look at this individual choanoflagellate, and you look at a choanocyte, you can see an obvious similarity between the two. So here's this ancestral choanoflagellate, gave rise to all the animals, really, including our first branch here, the poriferans. So here is our big phylogeny of animals that we're going to pick apart the rest of the semester. Here's our first and oldest branch, the sponges. Okay, so large range. Again, I showed you some pictures of sponges. Some are really small, some are huge in terms of the complexity and the size, the number of pores, right? And this is, we can kind of group those complexity patterns into asco, psycho, and leuconoid. Okay, the asconoid would be similar to those Grantius sponges we looked at in class. They're one single opening in the middle with multiple channels, small channels coming off the side. Right. Well, actually, no, there are no channels coming off of the side. The choanocytes are in the middle of the sponge. Psychonoid would be more, I guess probably Grantia would be a psychonoid. Single opening in the middle, but there are canals coming off the side that contain the choanocytes. And then these leuconoid have multiple large central openings with lots of incurrent canals, and these flagellated choanocytes in the chambers. Okay, and then the skeleton is made of collagen. Some have silica, and then there are the spicules embedded in that. Okay, so this would be the asconoid sponge. central spongiocele, central cavity, choanocytes lining that cavity. Here are the incurrent canals called ostium. Water comes in through the ostium, out through the osculum. Okay, so here's that cross section you guys looked at in lab, or you will look at in lab. Here's the spongiocele. Here are the side chambers lined with the choanocytes. Focus in, right? Water comes in through here, into the spongiocele, out the osculum. Here's those choanocytes. That would be more like a cycanoid sponge. Okay. And then we get into these leuconoid sponges, have multiple channels in the side, all lined with these choanocytes. Water comes in, move through here, filtered out in these individual side chambers, into the spongiocele, out the osculum. Okay, so these really, if you think about it, this is all about fitting more surface area and more choanocytes to do more filtering into a body. There's some spicules. Again, all is in the back are spicules too, and they're, depending on the group of sponges, they have different shaped spicules. So that is a taxonomic characteristic. Here's the spongin, the mesohyl, made out of collagen, which is a protein found in our skin and our nails and things. It's probably the most common protein in animals. Okay, sponge reproduction. Okay, we can fit this into both sexual reproduction. So in other words, it takes a male and a female, a gamete from a male and a gamete from the female are exchanged. Sponges can be what is called dioecious or monoecious. Two-ecious homes, two homes. So i.e. the male is in one home and the female is in another home. The male body is in this separate individual and the female is in this separate individual. Compared to a monoecious situation where we have one body with both male and female sexual organs. Okay, and actually sponges are weird because they can actually switch. So, you know, talk about gender roles here. Gametes are produced by the choanocytes. The sperm is released into the water. The female filters that out. She basically whips around with her choanocytes, draws in the water. The sperm is filtered out, fertilizes her eggs, and then sometimes the eggs can be retained. Other times, both the sperm and the eggs are just broadcast into the water. Think about the synchronization this takes. They have to do this at exactly the same time so that they will come together and form fertile eggs. The larvae is flagellated planktonic, kind of floats around and then settles down on the substrate. Or sponges, because they are so simple, have retained the ability to reproduce asexually. So you can actually slice a piece of sponge off and sort of plant it like a plant and it will regenerate. Okay, and they can produce these little kind of spores or cysts, these things called gemmules, that can sort of be placed on a pier or rock, and when the water dries out they are resistant, and then when the water comes back they can just form into a new little sponge. Here's some gemmules here. So they are all, these individual little gemmules are capable of producing a new sponge body, but which would be a clone of the original. It's not an example of sexual reproduction. So let me just show you this part of this video real quick here.
because in sponges, cells carry out all the functions that other organisms are taken care of by specific organs like the heart, or the nervous system, or the skeletal system, or the blood. So, sponge cells have many responsibilities to perform.
Unlike most of our cells, those of sponges retain a special freedom. They can continuously reinvent themselves, performing some truly miraculous feats. If a sponge is passed through a sieve, its cells are separated. But somehow the cells still recognize each other. Over several hours, they begin to come together, building many new sponges. No other plant or animal can resurrect itself this way.
Body.
Okay, so that the interesting part of that, if you think about it, as a pattern, these animals are very simple. They have very few cell types, but they are able to accomplish all the same things that our bodies do. They acquire nutrients, they digest, they distribute those nutrients, they produce waste to get rid of the waste, they produce gametes, they accomplish fertilization. So that tells you if there's only 3 or 4 cell types, each cell type must have more jobs, right? Those cells are not, they're kind of loosely multicellular. They are more independent than our cells are, right? So this is a sort of the early experiment of multicellular animals. In fact, you can take a sponge and blend it up, and the cells are independent of each other enough that they can just come back together and form a new sponge. Right? We can't do that. Why? Because each of our cells has differentiated labor so much, it has such a specialized job that without another cell type next to it, it can't survive. Right? So that is a uniquely sponge kind of characteristic here. They eat dead stuff and little stuff, detritus microorganisms. Um, these choanocytes both make the current and they filter the food out. The choanocytes ingest are phagocytize. Phagocytosis is basically where a cell envelops a particle. Its membrane grows around that particle and then it envelops. And the digestion of that food particle happens intracellular, inside of the cell. Very different. Think about our cells produce the enzymes which are dumped into the small intestine, and the food is digested outside of the cell. It is broken up into little pieces outside of any cell, and then the little pieces are then absorbed. Okay, so again, animals have a wide variety of ways to accomplish the same thing. Okay, so here are those choanocytes. Water is a current is created by these flagella moving, and we see as the water moves, little particles get trapped in these little microvilli or collar cell on the outside of the choanocyte. Okay, so here's a sponge. I recommend, well, let's just go ahead and watch this. It shows the same.
Most people, when they think of a sponge, they think on their bath sponge. And when you have a bath sponge in your hand, what you're holding is a soft skeleton that is made of collagen.
But unlike the bath variety, many sponges aren't soft at all. In addition to the soft collagen of the bath sponge, many have millions of brittle crystalline spicules embedded in their bodies. Spicules are microscopic structures that help give a sponge its rigidity and form.
Spicules are an incredible tool for us today to identify most of the sponges that are living in the oceans.
For most sponges, the glassy spicules act as a kind of fingerprint. Magnified, their dazzling array of shapes and sizes are as beautiful as they are unearthly. If the sponge is indeed an animal, it must eat to survive. How does it feed itself when it has no obvious mouth?
A sponge is actually a fantastic pump which requires an incredible coordination of cells to function. It's a living filtering machine. It lives by soaking in water and filtering out food particles.
These seemingly motionless creatures are working extremely hard in order to feed.
Imagine that to get an ounce of food, a sponge has to pump over a ton of water through itself. Now, can you imagine doing that yourself? Drinking a ton of water to get an ounce of food to your body?
This is cool. This shows how much water they
Diaz injects a
filter through.
harmless colored dye into the water near the body of a sponge.
There's those incurrent channels all along the side.
One of the ways we can test for the rate of water flow
Some kind of large
sponge body
leuconoid
is to
sponge.
inject a colored dye and measure the speed
Clanocytes
at
are
which
filtering,
it is
moving,
pumped through the
and then the
sponge.
dye comes through into the sponge
When
seal.
I start seeing the color dye coming out of the sponge in less than 2 seconds from when it was applied, I could not believe my eyes. This steady and strong continuous flow of water continuously coming out of the sponge
And up out of the
was an
osteum.
incredible realization of the dynamic existence of this organism. It's an incredible sight to witness.
These ghostly exhalations are proof secret inner world.
So this shows sort of the inside view of those clanocytes.
Drifting along with countless single-celled organisms, we feel the tug of a current.
Some diatoms,
In the wonderland
choanoflagellates.
within, this sponge's cells all
Or
work for the
dinoflagellates.
common good, but they do it in wildly different ways.
Pump through.
As the current draws us on, we pass sponge cells that are making new spicules. The walls themselves are treacherous. They can simply engulf their unfortunate prey.
They just phagocytize the food without even filtering it out.
As we are drawn through ever narrower passages, the current slows until we burst.
Okay, so you see, here's those choanocytes. Here's the flagella. There's those filtering collar cells.
Into one of the sponge's millions of tiny beating
Get stuck on that
hearts.
collar cell
These are colonocytes, cells whose whip-like
And
flagella
just-
powers the pump.
engulfed.
The flow quickens again as it
All right,
whisks us
boom,
away
out into the spongocoel,
through the labyrinth of canals that lead
up, out of the osculum.
into the sponge's central cavity.
All right. Pretty cool. So, that's how they accomplish eating. Every animal has a different story for how they do this. Diversity of sponges. Kingdom, Animalia, phylum, Porifera class. Two main classes I want you to know. Calcarea, which are the calcareous sponges, which are marine. Calcium carbonate skeletons, or hexactinellida, like the one we saw at the beginning. These are the glass sponges that have silicaceous spicules. Okay. See, like this, these silicaceous sponges. Class Desmo sponginae is the biggest sponge, the third and the largest. 80% of species are found in here. They're all this big, complex leuconoid type. They are found all around the world and very commercially important. Still, a lot of sponges are harvested from the wild and sold, but then there's also this cytosine arabinoside, which is a chemical produced only by sponges, and it is a very potent anti-cancer treatment. Okay, so that's just another example of sort of a seemingly useless animal, and a little bit of study that's been done have found some really interesting chemicals produced only by that group of animals. Big sponges, little sponges. All right, 27 species of freshwater sponges. Here we see them. They even have their own commensal spongilla flies. These things, the larvae of these flies only lives in freshwater sponges. Right? I'd love to find one. I've never seen one. Attached to rocks, logs, plants. And they're filter feeders, of course. A lot of them have these green algae symbionts. So when you see this word, you're thinking sim together, like Sympatric. These two living things living together. What does the algae get? Habitat and carbon dioxide. What does the sponge get? Well, the algae takes the CO2, turns it into sugar, right? And produces oxygen too. Very cool stuff. So that's phylum Porifera, and everybody should watch this week, whether you are taking the test today or on Thursday.